Next one is call it as plant regeneration pathway. How the plant that can be able to form the new pathway for the development. Here we want to select the X plant. That X plant is nothing but whether it is a root, shoot, leaf, flower, seed, fruit, etc. We want to collect as a X plant. Here meristem as a X plant or shoot apex or stem, leaf, root. Sometimes we want to select a callus as a explant. Here the meristem as well as shoot apex that can be able to mainly used for the virus free plantlet production, that is apical meristem culture. Simply we said as apical meristem culture. Then next one is call it as stem. The stem is mainly meant for the containing the node, that node that can be able to mainly used for the multiple shoot induction. That multiple shoot induction is nothing but it is also one of the process of micro propagation. With the help of micro propagation we want to induce the root, that is called it as root induction. With the help of root induction, that root that can be able to give the plantlets. After the plantlets they can be able to done under the some of the condition. That process is called it as hardening. That simply we say transfer to the field condition. Then next one is callus. The callus that can be able to form a indirect embryogenesis. The callus that can be able to develop a embryo or organ directly means that is call it as a direct embryogenesis. But the callus that can be able to develop the organ, that organ that can have the capacity to develop a shoot or a root, that is call it as a indirect embryogenesis. Here the callus that can have the capacity to develop a shoot induction or root induction, then after that can have the capacity to develop a plantlet, or after that plantlet undergo a hardening, then the transfer to the field. Here the callus they cannot develop a embryo directly, so that is called it as a indirect embryogenesis. Here the explant that can have the capacity to directly develop a embryo, that process is called it as direct embryogenesis. That direct embryogenesis we will get the embryoid. That embryoid they can have the capacity to develop the plantlets. After the plantlets undergo a hardening process, then after it can be able to develop a plantlet. That plantlet that can be able to transfer to the field condition. This is the one of the flow chart for the plant regeneration pathway. Here the plant regeneration pathway, then they can be in a diagrammatic view. Here we want to select a well developed plant. Then afterwards we want to select the meristem or shoot apex, or we have to select a stem or leaf or root as a explant. Through that explant, the undifferentiated mass of unorganized tissue that can be able to develop, that is called it as a callus. Then from the explant, here the embryo that can be able to directly developed means that is called it as a direct somatic embryogenesis. The direct somatic embryogenesis that can be able to occur after the explant with the help of callus that can have the capacity to develop a root, or that can have the capacity to develop a shoot. That process is called it as indirect embryogenesis. Here in indirect embryogenesis also the plantlet that can be able to obtained. But direct embryogenesis we want to get a somatic embryo. Afterwards that somatic embryo they can be able to develop a plantlet. It is the diagrammatic view for the plantlet formation. It is the diagrammatic view for the plantlet formation. Then next one is call it as what is somatic embryogenesis and what are the applications for the somatic embryogenesis. Then here what is somatic embryogenesis? Simply we say Somatic refers to body cells. With the help of body cells, the embryo that can be able to develop. The process of getting embryo from the body cells, that is called it as plant body cell, that is called it as somatic embryogenesis. Afterwards, the somatic embryogenesis is the formation of embryo from the callus tissue directly, and these embryos are called embryoids. The callus tissue that can be able to develop a embryo, that embryo is called as embryoids, that can be able to occur in the in vitro condition. In vitro cells directly form the pre-embryonic cell, which differentiated into embryoids. Simply I explain. Somatic embryogenesis is nothing but process of getting embryo from explant directly. That process is called as somatic embryogenesis. Through this somatic embryogenesis, we get a embryoid. That embryoid that can be able to occur with the help of in vitro condition. That in vitro conditions that in vitro condition, that embryonic cells, they can be developed from the pre-embryonic cell. That pre-embryonic cell that can be able to differentiated into a embryoids. Then next one is called it as application. What are the applications that can be able to present in the somatic embryogenesis? Somatic embryogenesis provides potential plantlet. The potential plantlet means viable plantlets. When the viable plantlets that can be able to get means, after hardening process only we have to obtain the viable plantlets that can be able to established into a plant. That plantlets after the process of hardening that can be able to develop a plants. Then next one is used for the production of synthetic seed. The production of synthetic seed is nothing but production of artificial seed. What is synthetic seed? I explain later. Then next one is called it as somatic embryogenesis is now reported in many plants such as, for example, Allium sativum, garlic, vellapudu, Hordeum vulgare, that is called it as barley, Oryza sativa, paddy, nillu, Zea maize plant, Makachola, and this possible in many of the plants. Here synthetic seeds also produced by encapsulation of embryoid in agarose gel or calcium alginate. Synthetic seed is nothing but artificial seed. The artificial seed that can be able to obtained from the encapsulation of the agar, that is a agarose gel. Instead of agarose gel, we want to use the calcium alginate. Then synthetic seed is nothing but a artificial seed. Then next one is called it as organogenesis. What is the meaning of organogenesis? The process of development of organ, that is called it as organogenesis. Simply, the morphological change occur in the callus leading to formation of shoot and root is called organogenesis. The process of the development of shoot and root that can be able to develop from callus in morphologically, that is called it as organogenesis. Here the organogenesis that can be able to classified into two main category, that is called it as calogenesis and rhizogenesis. What is calogenesis? Callus that can be undergone a differentiation that can have the capacity to develop a root. Sorry. The callus that can be able to differentiated and it can be able to develop a shoot. That shoot they can develop a plantlet. That process is called as calogenesis. Calo refers to callus. From the callus the shoot can be developed, that is called as calogenesis. Next one is called as rhizogenesis. Rhizo refers to root. The root that can be able to develop from the callus, that is called as rhizogenesis. With the help of root that can be able to develop into a plantlets. That process is called as rhizogenesis. Organogenesis that can be able to induced only in the in vitro by introducing the plant growth regulators in MS medium. The plant growth regulator is nothing but auxin and cytokinin. The auxin is meant for shoot formation. The cytokinin is meant for root formation. Auxin is meant for shoot formation and cytokinin is meant for root formation. Here the organogenesis are of two type. One is calogenesis and another one is rhizogenesis. The shoot that can be able to arise from the callus, that is called it as calogenesis. The root that can be able to arise from the callus, that is called it as rhizogenesis. After that, the plantlets that can be able to obtained. Thank you.